Thank you. It's my great honor to be here participating in this conference. I would like to talk about many non-human primate research. I'll be on the screen. Before I do that, I thought about this new technology, what potential impact to the society. Mute. Yeah, this is muted. Yeah. Well, I think it's bad. There's using the Zoom, so that's a problem. Maybe we should just ask them to go on the cut off the. Okay. There's still brief. Okay. Now it's better. For the neurotechnology, I think we have brain-machine interfaces for monitoring, decoding, manipulating brain activity, which I've heard some Ed has talked about. We also have drugs that can repair or augment brain functions. I think that's a new technology. We also have virtual reality, metaverse, GPT tools now for sensory and cognitive manipulation. In fact, I would consider this as a new form of neurotechnology that would affect the brain greatly. Now, we're probably not going to discuss this in this meeting, but there are many genetic and cell biology techniques for brain disorders, developed for brain disorders, including the early diagnosis of brain disorder. There are privacy issues, genetic screening, all this would be involved in the ethical issues. There are cell replacement technologies, stem cell therapies, trans-differentiation, or just induced differentiated cells implanted into the brain. There's also gene editing of the brain tissue of the embryo. We're familiar with that. The area I'm going to focus on is research using non-human primates that involve gene-edited or treatment-induced models of brain disorder, and also human cell and tissue xenografts in non-human primates, human tissue introduced in non-primate. There are potential ethical issues involved. There's production of stem cell-derived monkey chimeras for disease models. At present, animal chimerics are being produced. I'll show some example of the monkey chimera. But in the long run, when we talk about human monkey or human pig or human animal chimerics, that will be going to be ethical issues to be involved. So let me begin with the editing of monkeys by transgenic approach. Usually, we have an in vivo fertilized embryo, injecting sperm into a mature oocyte. We get the embryo developed and then inject editing tools like CRISPR-Cas9 editing tools to knock out the gene or editing the gene. And then that embryo developed in culture and then implanted in the surrogate mother to give birth to the monkey. Now, this is the usual way of generating embryonic editing to generate monkey transgenic monkey edited gene edited monkey model. So I'll show you some example. The most successful example is in editing of circadian rhythm gene. Now, circadian rhythm is very important. It controls many of our metabolism, daily physiology. It involves a cycle of a gene product, daily cycles, controlled by a few core genes, like female 1, clock gene, period gene, pride gene. Although these are well-known genes, their level goes up and down during the daily cycle. Now, one of the core genes called BML1, which controls the expression of period gene, which triggers a lot of downstream gene, hundreds, if not thousands. And then they also inhibit reduced level of BML1 to pull it down during the night. So you have a daily cycle of these transcription factors responsible for controlling large number of genes. Now, we thought this circadian gene is good because they are involved, their disorder or their mutations are involved in premature aging, in diabetes and even in cancer, in neurodegenerative disease. So presumably it's an upstream gene that affects lots of downstream genes, affecting many, many brain processes. So I think if we lock that gene out, maybe we see a lot of brain disease phenotype, rather than, because most of the gene phenotype, as we discussed, is polygenetic or in origin. So Hongjin Chen in our institute was involved in this work. He knocked out, using CRISPR-Cas9, editing one of the exons of the BML1 gene, knocked that gene out, and then he produced 3 complete knockout monkeys and 2 mosaic monkeys with partial knockout of BML1. The complete knockout BML1, there's no BML1 in the body. The partial knockout, you have reduced amplitude of oscillation of BML1 and with you reduce level. And the effects are very clear. First of all, the neuroendocrine in the bloodstream, in the serum, is usually in wild-type monkeys oscillate during the day and night, like melatonin goes up during the night, goes down during the day. Testosterone, DHEA, cortisol, and all have these daily cycles. In the complete BML1 knockout, cortisol levels remain high. Now, cortisol is so-called stress hormone. So we expect that the monkey will have high stress. And melatonin are reducing and very low level, oscillation reduced. Sleep disorder. This is all expected phenotype. In fact, we do show that the sleep, both REM sleep and non-REM sleep, are greatly reduced in these knockout mice or in partial knockout mice. And this reduction of the sleep is enhanced if you continue 3 days without 3 day complete night, 24-hour night over 3 days. You can see clear difference. Wild type and control, and a knockout monkey show greatly reduced non-REM sleep and REM sleep. So this is what we expected. More interestingly is these knockout monkeys show stress phenotype. They don't move around as much as control monkeys. They confine one corner of the cage. And all the parameters indicate that both the complete knockout or partial knockout shows reduced motility during the day, coincide with the sleep disorder. And also, interestingly, they show a clear phenotype in terms of anxiety. When a normal monkey, when there's a feeding time comes, it comes in front of the cage and interact with people. The disease, this knockout, BML1 knockout monkey are hiding away from people, shy away from people, always hide in the corner of a cage. This is very typical for both, with a different stage, different degree of phenotype. Complete knockout shows most severe phenotype of stress or anxiety or depression. Now, there's an interesting EEG recording methods for detecting schizophrenia patients called mismatch negativity waves. In EEG pattern, you can detect when you give a sequence of sound, and it comes with a deviant sound, beep, 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 beep. When the deviant sound comes, you see this wave, mismatch wave, in a human with schizophrenia compared to control. The control, this wave is not as prominent. Monkey, the BML1 monkey shows very prominent mismatch. So this is again an interesting phenotype, coincide with a human phenotype. But all these monkeys are heterogeneous, different degree of phenotype, presumably because their genetic backgrounds are different. So we thought that many years ago, like now, 10 years ago, we thought the best way to produce monkey model is to clone the monkey, to have a uniform genetic background, and then you have you can use that as a test. So cloning of primate species has been difficult since the dawn of the ship. 20 different mammalian species have cloned, but by 2012, 10 years ago, monkey cloning has been not successful, primarily because of the somatic nuclear transfer. The somatic nucleus many of their developmental genes are suppressed. So you transplant into an oocyte, they cannot substitute trigger the development properly. Now in mice, in other species, this can be overcome for some reason. But in monkey, this has been difficult. But over the past, by 5 years ago, we were able to overcome this by the use of epigenetic modulator. So the procedure is standard. You take a cultured fibroblast from a monkey, you culture a large number of proliferating fibroblasts. You fuse the fibroblast with an oocyte with its nucleus removed, right? So you have a nucleated oocyte. You fuse that, you get a fusion reconstructed oocyte with somatic nucleus. Now, you wanted the nucleus to develop. So to do this, we tried a large number of epigenetic modulator. Eventually, this histone demethylases together with histone acylase inhibitors works well. It triggers most of the gene, developmental gene, and that embryo developed well in surrogate mother and give the clone monkey. So 2018, we announced the clone monkey, two monkeys, sisters, from fibroblast of a female fibroblast from a female host. Now once this cloned, I thought, well, we have this female white knockout mice, which has a very clear phenotype of depression and anxiety. We should clone that monkey to see, so that's what we did. We cloned this monkey with the phenotype, with this depression phenotype. We cloned 5 monkeys out of that. These monkeys show a very clear phenotype, very close to their fibroblast donor. So they are a few weeks after birth, in the incubator, they start to show this depressed phenotype. We are now using this group. We have 10 or 15 cloned plus genetically modified female one modified monkeys. We are doing drug tests. Some candidate drugs for depression. This small molecule drug were used to give orally to the female one knockout monkey. Now, this monkey usually, before treatment, they are afraid from people, will not come over to grab the food. After the treatment, after oral intake, they are very normal compared to the wild type. And when the drugs wears off a few weeks later, the phenotype return. Right? So this is a clear sign that only a few monkeys group in 5 or 6 control, 5, 6 experiment show very clear phenotype. Here's a social defect. The knockout monkey usually refrain from, before treatment, refrain from interact with the wild type monkey or other knockout monkey. So there's a middle cage, they come to play, come to interact. They tend to confine in their own cage. But after taking the drug, this candidate depression drug, they start to work play in the middle. So the social interactions is increased. Here is this TPN, this candidate drug. They increase the interaction. Compared to another depression drug under clinical use, the effects is better. Melatonin has no effect. Melatonin helps this monkey to go to sleep but they have no effect on the depression phenotype. Okay, so by this approach, we have genetic edited a large number of monkeys. Here are some of the disease we are working on. The most successful gene editing monkey is the autism phenotype. In particular, the Angelman syndrome, which has a very clear genetic origin, genetic cause of UB3A protein, AE3 ligase. This knockout of this UB3A, in particularly in the maternal copy of this maternal allele of UB3A, if you knock that out without affecting paternal copy, you get Angelman syndrome-like phenotype with decreased motility, motion, and reduced cognitive ability by the standard Wisconsin CAR test, and also social interaction. Right? These monkeys show this 2 years old monkey of this Angelman syndrome phenotype. There's also another Fragile X syndrome, part of a subtype of autism, with a knockout of a particular protein known to be related to Fragile X syndrome, FMR1 protein, also show reduced social interaction as well as the cognitive. They show cognitive impairment by standard test. There are motion, the dystonia motion, monkey, epilepsy phenotype. We try very hard to do AD and PD phenotype by introducing human mutation, APP, PS1, tau protein, human protein with mutation into transgenic into the monkey. We have monkey born, they are 1 year old, have not shown any phenotype. PD monkey as well. We have both Harkin knockout as well as mutated alpha-synuclein protein, human protein introduced into the monkey. They are born, but we haven't shown phenotype. Now we expect this phenotype will come later because this is a neurodegenerative disease. Another way of producing monkey model is induced or spontaneous monkey models, which we can induce addiction model, similar to that in the mice and rat. Self-administration, the monkey press it on the liver, you will get an injection of methamphetamine into the bloodstream. And when they know, after a while, they know that they enjoy doing that, and eventually they got addicted. The addiction show after you terminate the drug. So there's a standard addiction model. There's an epilepsy model by drug-induced epilepsy or electrical stimulation induced drug epilepsy. And most interestingly, in large colonies of monkey, we screen out OCD phenotype. A few monkeys, we have 15 also, out of a few thousand monkeys. They show a spontaneous OCD phenotype, which shows motor behaviors without stopping, high frequency over long duration. Now this monkey, if you look at their SNPs, the genetic gene makeup, there are SNPs in these monkeys different from the wild-type monkey that are similar to human with OCD. So there are some overlap of the SNP gene genetic node site mutation that are similar. So this spontaneous mutation could be a model for study of neuromodulation or drug therapy test. Now this all for development of these monkeys for clinical use, potentially for preclinical testing of therapeutic approach. And there's another group of experiments which we think that would be more interesting to discuss. Those are the study for basic neuroscience, for cognition. That is to introduce into monkeys that change the monkey development to understand how human specific gene might play a role in the human brain development, cognitive development. Now there's a there's a MECPH1. This is the microencephalin gene. A mutation of this gene causes a small brain. So normal human MECPH1 gene, if you put into a monkey, transgenic, it will cause delayed neuron maturation, similar to the delay of neurodevelopment in humans, so-called neoteny. Now, human-like expression delay also found in the synaptic level, in the neuronal differentiation and synaptic signaling gene expression. And if you look test the behavior test is rather limited, but so far, the short-term memory test, the transgenic monkey that develop their brain, with their brain developed slowly like humans, have higher, had a higher ability of performing this working memory test, right? Delay match to sample time. And there's another recent paper just impressed, also from the same group, taking a human-specific duplicated gene into the monkey. The bone monkey had, if you show the MRI images, actually show with time, longitudinal imaging show delayed brain development also, with region-specific volume changes accompanied by changes in myelination level in temporal occipital region. There's this also inhibited increased deep layer neurons during fetal neurogenesis and delay synaptic maturation in adolescence. Interestingly, this monkey also shows improved motor planning and execution skills. So now this is controversial because this you are putting human gene into monkey, not for curing disease, but for studying how a human ability comes about, how human brain comes about. People, when the first paper comes out, the media there was a media praise about where these groups are creating monkeys, creating as a planet of the ape type monkeys, monkey with intelligence. Because all they show is a delayed development of the that's been caused by this gene. If you do this experiment in mice, it's okay. So far, it's okay. But in monkeys, here to give people this idea that it's dangerous to do. I mean, this is one for discussion. I think many of the frontier work in primate, non-human primates would involve the territory, because the reason of doing non-human primate to study them to understand our human. Now, the way to do it is to try to see what genes in the human-specific gene development can play a role in a specific aspect of the monkey brain. Then this experiment is a good experiment. I mean, for scientists, it's a very important experiment. So finally, before I end, I just talk about this most recent development. All this, the cloning, transgenic approach, cloning approach, as Anna has talked about, we need to have an effective way of transfecting the cells in a large number of tissues. And this cloning and embryonic manipulation are inefficient. The efficiency is low. So the best approach, following that the approach in mice, is to do chimeric, produce pluripotent stem cell in culture, grow in large number, proliferate in large number, inject this stem cell, or you do manipulation of this stem cell and screen out the manipulation you do, genetic editing or whatever, and then inject the desired stem cell into the embryo to produce a mosaic embryo. And the mosaic embryo, as long as you cells derived from your stem cell is large enough in number, then you can affect the tissue function. You can do experiments on the animal. So this can be tested by all the molecular tools, gene testing, to see whether you really have the donor stem cells surviving in the adult, in the tissues of the mosaic animal, chimeric animal. So then you have accomplished this 3 weeks ago. He published this paper showing the very high percentage of incorporation of stem cell derived cells in the chimeric. Now you can show that, particularly in the brain, if you do all the cells, you do cell gene profiling of the cells, it goes into the stem cell, GFP marked stem cell, found in all the cell types, essentially, in the brain. 90% of the tissue cell, brain tissue cell, are derived from the stem cell. So this is useful, right? One can think about manipulation. Right? And also, it goes into the reproductive system. This fibroblast comes from a male, so the spermatogonia, the precursor sperm cells are derived from the donor cell. So you can talk about germinal transmission like in the mouse. You can essentially reproduce these animals. So I should stop here.